What's up, everybody? We all have that one friend that helps keep us calm when the world starts to drive us completely insane. Or maybe you are that friend. Now, in this video, we're going to be talking about everything that you need to know for C4.1. There's a ton of content, so around midway, I'm going to take a mental break. I'm going to get a coffee, do whatever. Maybe you can do some push-ups, grab a pizza, whatever helps to reset your brain. And at this point, we're going to do some IB style questions, and then we'll continue with the rest of the content. And at the very end, we'll do a couple more IB style questions. Now, before we start, go check out teachme.org for some mock exams, tons and tons of IB style questions, and the most beautiful notes you'll ever see. Now, without further ado, let's just get started. You may have noticed that the title of this chapter is Populations and Communities, right? So naturally, we're going to define these guys, but I find the best way to define them is by starting off by defining species. So here we have a nice environment from Lion King. I don't know if you've watched that movie, but let's define species. What makes two of these organisms in this environment different species or the same species? Well, for two organisms to be the same species, they must be able to reproduce and make an offspring. And that offspring must be fertile. For example, we know if a lion mates with another lion because they're the same species, they will be able to make an offspring. And we know that offspring will be fertile. That offspring can go and find its own mate and have babies. But we know if we take a lion and let's say a tiger or a giraffe, they will not be able to make an offspring. And even if they could, by some miracle, make an offspring, that offspring will be infertile. That offspring is called a hybrid, right? And they will not be able to have babies. So therefore, those two organisms were not the same species, right? So that's what you should know about species. So a group of lions, so we can see in this diagram, there are a ton of different species, right? Lions, giraffes, whatever, a lot of stuff. So what's a population then? A population is a group of the same species. So this right here would be a population of giraffes. That right here would be a population of zebras or maybe elephants, whatever. So a group of the same species is a population, okay? What about community? So a community is even one step further. A community is a group of different populations. So this right here is pretty much a community. We can see a population of giraffes. We can see a population of elephants, population of zebras. This right here is a group of different populations living together at the same time, right? And interacting with each other. That's a community. Now, what about one step further? Ecosystem. An ecosystem is a community. So all these organisms and populations interacting with each other, and the abiotic environment, the non-living environment. So I'll give you some examples of what I mean by the abiotic environment. That would be anything that's non-living. For example, sunlight, water, these stones, anything like that is abiotic. Okay, here are some examples. When we say the word interacting here, that is a very broad term and it doesn't mean anything specific. Interacting can mean killing each other, when the different populations kill each other, when they help each other, when they compete with each other for resources. All of those are different forms of interacting. So just know that interacting is a very broad term and there are many forms of interacting. And we're going to talk more about that later in this video. So now let's take it one step further with defining populations, because we know, right, a population of giraffes in Africa is not the same population as a giraffes living in America, right? Because we know, according to the definition, to be classified as the same population, you must live in the same area at the same time. So naturally, if there's a population of giraffes in America, then they're not the same population. Now, that brings me to the question, where do we draw the line then? How far apart do they need to be for them to be different populations? What exactly is the measurement? Well, there's no measurement, but we can answer it like this. Based on whether they can interbreed or not. If one group of giraffes will never interbreed with another, we say they are different populations, okay? So that's how you define different populations, based on whether they will interbreed or not. So if we look at this example here, there's one island here that has a bunch of penguins. let's call them population A, and another one here that's population B. We say they are different populations, A and B, because they are isolated. They are so far away from each other, they will never swim across to the other land. It's way too far. And therefore, this one will only interbreed or reproduce with its own people here, and this one will only interbreed or reproduce with its own people here. They are isolated reproductively. They are isolated. Only these will reproduce together, and only these are reproduced together. We call that reproductive isolation. And because of that, because they won't interbreed, they are considered different populations. I hope that idea makes sense. This brings us to carrying capacity. What exactly is carrying capacity? So we know each environment can only hold a certain number of organisms. They can't hold an infinite amount, right? So that is exactly what carrying capacity is. It is the maximum number of organisms that a particular environment can sustain or support. Now, scientists like to represent carrying capacity by the letter K. Who knows why, but they want to, okay? So just remember, K is the symbol for carrying capacity. Now, why does an environment have a carrying capacity? What causes there to be a carrying capacity? Well, limiting factors. So, limiting factors are factors that basically prevent certain organisms from surviving. Okay, it's a factor that limits the survival of an organism. So, because these factors limit survival of an organism, therefore they limit the survival of the population, and therefore they determine or they define the carrying capacity, the maximum number of organisms this habitat can sustain. So we're going to we can classify these limiting factors into two broad categories: density-dependent factors and density-independent factors. So we're going to use some examples to illustrate this, but I have a nice way for you to remember these. Panda, paw. Panda mnemonic will help you remember the density-dependent factors. Paw will help you remember the density-independent factors. You don't have to remember this mnemonic. This is just a way for me to help you out. You can just remember the individual things if you want to. You don't need to know the mnemonic. Okay, so just know that. So what are density-dependent factors? Density-dependent factors are these factors that affect the population more as the population number increases. Whereas density-independent factors, they affect the population no matter how big or how small a population is. So let's take a look at some specific density-dependent factors. The first one we're going to look at is predators. Take a look here. Predators. This is going to be our chicken. We know it's a prey, right? Now let's say it lives on this island. And let's say there are predators. Let's say a human like Maui. And he catches chickens because it's his breakfast, all right? Now we know that when this chicken population is very small, like for example only a few of them on the island, then they're going to be very hard to catch for the predator, right? You can imagine, like, I'm pretty slow. I don't know about you, but if you try and catch a chicken, they're pretty damn fast, and they're just going to run circles around you, right? They're hard to catch. Therefore, it's hard to reduce their population number. But if they reproduce and their population starts to increase, then what do you think is going to happen? Will it get easier or harder to catch them? It's going to get way easier. So as the population grows, right, of these chickens, their density increases. So they're getting more densely populated. And when that happens, a predator, they're basically going to be all around the predator. The predator doesn't even have to go and chase them. They're right next to them. They're pretty much so dense. So in this scenario, the predator has a very easy job to catch this chicken and reduce its population number. So therefore, this is considered a density-dependent factor because this factor influences a population more as the numbers increase. More the dense they get. So it depends on density of the population. The less dense, the less it will affect the population. The more dense, the more it will affect the population. Let's take a look at another one. Availability of resources. So this is a pretty good one. You can think of the chickens here. The more their number, the higher their population density gets, the more scarce food will become, because they all need to eat breakfast, right? Now, if they all have to compete for food, it will get worse and worse the higher their density is, because food will remain the same, but their number, the number of people who need to eat, will get more. So therefore, all of these things will influence them as well, the more dense their population gets. And another one I can use that I was going to share earlier is forest trees when they grow, because they compete for sunlight, right? Now, when there's only one tree, it's fine. It's got all the sunlight for itself. But if the trees get very, very dense, imagine it takes up this whole space, okay? Now, if you get a seedling, let me show you. Now, if a new one comes, a seedling, guess what? Now, all these trees around it, because the population is so dense, is blocking the sunlight, preventing it from getting sunlight, or even space, or even soil, because they're all taking up all the space. Therefore, this one will not be able to germinate and grow into a full-grown tree. The only way it can do so is if one of the parents die, and now it gives a vacant spot for it to be able to grow in and absorb sunlight. So you can see at this point, the only way a new organism can arise is if an old one dies. So at this point, you're at a sort of plateau. The population number is no longer getting bigger. All that happens is one will die and one will grow. So it will stay at a stable level. All right. Next one is nutrient supply. So you'll notice this mnemonic, some things overlap a bit, so it's not perfect, but it's going to help you out regardless. So when I talked about resources, I was specifically referring more to things like space and water and sunlight and oxygen and things like that. But the same thing goes for nutrient supply and food, as the example that I just mentioned before with the chickens. Now, what about disease? Do you think disease spreads more easily if a population is small or if the population gets bigger? The answer is as the population gets bigger. We know COVID, right, is a contagious disease, and it will spread way better in a population that is way more dense because the people are always getting in contact with each other. If the population was more small, the individuals would be sparsely distributed, and to spread the disease would be a bit more difficult because people don't come into contact as often as a very dense population. Next one is accumulation of waste. I'm going to use a fish tank to illustrate this. When you have one fish in a big tank, it will poo-poo and pee-pee and it will make waste, right? And waste can be toxic if it's very highly concentrated. But if there's only one fish in this big tank, then the waste is not going to influence it, right? It's not going to limit its survival. But if you now have 100 fish in that tank, now they're all going to pee-pee and poo-poo, and that waste will accumulate way faster. And so guess what? Now this is why it's a density dependent factor, because if there's 100 fish, the more fish there are in the pond or the more fish there are in the tank, the more quickly it will get dirty, full of all this waste. And waste can kill the fish and limit their survival. All right, so those are some density-dependent factors that you need to know about. Let's go on to the other side, density-independent factors. Before I do that, I can say here, so you can see that density-dependent factors keep a population at its carrying capacity. It is the reason why these guys are not going over their carrying capacity, because as soon as they get to a big number, their number will get restricted by all of these factors, keeping them at a carrying capacity. Okay, so we call this, right? What do we call this idea when you prevent a number from going out of control? We call that negative feedback. Negative feedback is what these weapons, these density-dependent factors are really doing. When a population is getting bigger, these guys are acting on them by negative feedback to reduce their population from going too big and returning them to normal range. All right? That is the opposite of positive feedback, which is a feedback system that reinforces or amplifies a response, making them more and more faster. And we're going to talk about this later in this, both of these feedback systems later on in this video. So, an example of a density-independent factor, so a factor that influences the population regardless if they are small or big, would be phenomena like natural disaster. Imagine if there's a forest fire here. All of these trees, regardless if there are 100 or 1, they will get burned down. They will get affected. It doesn't matter. It doesn't get worse if they are more or less. Regardless, they will be affected. Abiotic factors. So these can be things like temperature or carbon dioxide levels. So for example, I'll talk about carbon dioxide levels. We know as environmental pollution increases, so carbon dioxide levels increase, they will start to deposit more and more in the oceans. And this causes the oceans to acidify because carbon dioxide is acidic. Now, an acidic ocean is bad for our corals because it causes them to start dying. Our coral reefs is bad for some of our fish. Okay, so this is a density independent factor because no matter if there are 100 fish in the ocean or 1, ocean acidity will affect that fish regardless. Okay. Last one, weather conditions. So for example, floods, hurricanes, climate change, storms, all of these will also affect the population, again, the same way, no matter its density. Now, you'll notice that some of these factors could overlap in a different category, so that's why I said before, it's not a perfect mnemonic, but it's a way to help you remember when you have 10,000 other things to remember on the test. Now, one important thing I want to point out about density-independent factors is that they don't work exactly the same as a density-dependent factor that does it by negative feedback to reduce it from passing this carrying capacity. They kind of work very suddenly. So these guys will suddenly decrease the population by a lot. All right, so they work more in a harsh way, and they don't always hinder a population. For example, if there is a flood, yes, that will be very bad for certain populations that can't survive in water, right? So if it wipes away your house and now you can drown. But it will be very good for fish who live in water. Now they have a lot more water in their environment. And a lot more environment to live in because the water is now dispersed in a wider area than before. So these can help or hinder the population. It depends on what species perspective we're looking from. So that's what you should know about carrying capacity. Now that you understand that population numbers can be controlled by all of these factors or influenced by all of these factors, we need to learn something else here. What do we have here? We have a food chain, and we learned all about this in C4.2. So what's happening here is the arrows are showing the direction of energy flow. So when a cactus gets eaten by a jerboa, some of its energy will flow to the jerboa. And when the jerboa gets eaten by a snake, some of its energy will get transferred to the snake. So what we need to know about this whole concept here is any population, their number can be influenced from one of two directions, either from the higher trophic levels, so by getting eaten, that can reduce their numbers, or from the lower trophic levels, when their food gets more scarce, because if their food is scarce, then they cannot eat and they cannot grow their population or they might die, right? So there's actually a term you have to learn for this. So the first one is top-down control. So the first one that I mentioned was a snake's population number can be controlled from a higher trophic level by being eaten by these higher trophic level organisms. So that is an example of top-down control because you're controlling the snake's population from the higher trophic levels. So this can include basic things like predation and herbivory. So even the cactus's number can be controlled from a higher trophic level by the jerboa who eats plants. That's herbivory. So the opposite one was bottom-up control, which is just when your food is getting scarce, your resources are lacking, and that is causing you to die or not be able to survive, right? All right. Now I want to quickly show you something to illustrate if you really understand this. Let's say hunters go and hunt phoenix foxes and really decrease the hell out of their numbers. How is that going to influence the rest of the food chain's number? So this is going to be an example of top-down control because we are now reducing a higher trophic level organism's number, just like this. You see, we're reducing their number. This will then mean there is less top-down control for the snake. So the snake's numbers will now start to increase because there is less predation. And if the snake's number starts to increase, that will be a bad night for the jerboa because now there's a lot of snakes to eat jerboa, so their numbers will start to decrease. And then again, if the jerboa's numbers start to decrease, then the cacti will be happy because they can now grow again very, very easily. So you can see it alternates from top-down control. What about bottom-up control? Let's say we reduce the number of cacti. Say the weather conditions are very bad for the cacti and they start to decline in number significantly. Maybe the sunlight is very inadequate. If their numbers start to decrease, now what's going to happen to the jerboa? Well, it's going to decrease because its food supply is lower. And that means the snake's food supply is lower, so it will also decrease. And that means the phoenix fox's food supply is lower, so they will decrease. So notice that there is a difference between the outcome in the food chain, between the bottom-up control and the top-down control. So that is all that I want you to know about the top-down and bottom-up control of a population. With what we understand now, learning the population growth curve will be super simple. So I accidentally mixed these two up. We're first going to learn about this one, and then we're going to go over and learn about estimating population size. So here is the curve we're going to look at, and don't have your mind shut down just because we're looking at something mathematical right now. It's just a graph and it's not that bad, especially after we break it down. So you can see here the x-axis is going to be time, so as time goes on, and the y-axis will be population number. So the higher we go, the higher the population number. And now we have this K. Remember K, I told you, was like so weird because they give you a symbol to remember it with. It's the carrying capacity. So this line right here represents the maximum number of organisms an environment can, particular habitat can support. Now, this growth curve will have 3 distinct stages, 1, 2, and 3, and we're going to break them down stage by stage for it to make complete sense. The population growth curve will have an S shape, and we're going to understand exactly why it has that S shape if I do a good job at explaining it. So this curve won't be linear, it won't be exponential, it'll be S-shaped. So let's do it. Let's break it down. First stage first. Initially, any population's number is very low in the beginning of time, and then they will start to increase, and then it will start to increase faster and faster and faster and faster and faster. So it's exponential, this first part. That is why the first part of this curve, stage 1, is called the exponential phase, or also known as the logarithmic phase. Both names are the same thing, mean the same thing. You notice this is an example of positive feedback because there is a change happening, right? There's an increase in population number, but then that change gets amplified. It gets reinforced. So that change happens faster and faster with time. Do you remember when we talked about it here? That's what positive feedback was, right? Now the question is, why is this happening? If we go back to our chicken example here, when there is very few chickens, so imagine there's only a couple of chickens here. There is very little effect. They are very little affected by these density-dependent factors because their population is very sparse. They're not dense yet. The population is very small. So because these density-dependent factors don't influence them yet, they can easily find food and they can easily reproduce. So because they can easily form offspring, that will cause the population to grow a little bit. But if they have 10 offspring, every single one of those offspring can now start their own life and have their own kids. So if each of these kids now have their own kids, then the population number will increase dramatically, exponentially, right? So that is the mechanism why the initial part of the growth curve is exponential, okay? So I already said kind of the reasons why. Because of basic things like there's little density-dependent factors, right? The predation isn't affecting them much because their population number is very sparse. Same with the resources and other abiotic factors. All of these density-dependent factors that I mentioned before. Now, that's the first part of this curve. We need to look now at this little scale. So what's really happening is the reason why the population number is increasing is because of 2 combined things. Firstly, births. I just mentioned births. We call that in science natality. So another name for birth in science is natality. But that's not the only reason why a population number may be increasing. It could be because of immigration. If there is a penguin that's coming from a distant land and it decides to join this population, that has now increased that population's number, right? So both of these 2 things combined influence increase, adding to a population. Now the opposite things will contribute to removing. So deaths, as if a penguin dies, it will get removed from the population. And same with exiting a population, when one decides to ditch that population and go live its own lonely life, all right? So at the moment, natality plus immigration is more than deaths plus immigrations, much more. Following that, we have now our second stage. Notice what's happening here. We are now transitioning from a positive feedback system to a negative feedback system. So we're transitioning from an exponential sort of growth to a growth that slows down over time. That is why this curve actually looks like it has an S shape. So because it's transitioning, we're going to call this a transitional phase, right? Transitional phase. And it's because of this negative feedback system. So how does it work? Let me show you. Remember our chickens, when their population gets very denser and denser and denser, what does that mean? Then we have these density dependent factors, all of these weapons, preventing it from increasing. Doing negative feedback, preventing it from going beyond the carrying capacity. All of these things that we talked about before. So these things will prevent the growth, and that's why it's slowing down with time, preventing the population from getting too big. All right? Now, let's look at this guy here again. So now you'll notice, now the adding is not as extreme compared to the removing. Now they're getting a bit more even. The population is still growing, right? But very slowly now, okay? And that's because our natalities are not as high anymore, and our deaths are not as are increasing a bit because of the negative feedback system. The final stage looks like this. You'll notice that the population curve number now reaches a plateau. That is why we call this stage the plateau or stationary phase. So the population number will now stabilize at the carrying capacity. And the reason for this is negative feedback. The same reason as for why the population was decreasing in step 2. Because of all those density dependent factors reducing it and keeping it there at the carrying capacity. Okay, so it's the same reason. We're just now at the very extreme, right there at the carrying capacity. So that's all you guys need to know about the, oh no, not yet, almost. Remember this little scale? Now they're exactly even. So the adding to the population is now the same as the removing. Okay, that's why it is plateauing, not increasing nor decreasing. Finally, we get to this part here, estimating the population size, and then we can then we're going to do a couple of IB style questions and have a coffee before we continue to the rest of the information. Okay, so some scientists, or a lot of scientists, part of their job is to be able to count population numbers, the size of a population, the number of individuals in a population, because they need to know that when they try and figure out trends over time. How is this population's number increasing or decreasing? Are they gradually going extinct, or are they thriving? Is climate change affecting this population's number? And they can do many other ecological studies and things like that. So for many reasons, they need to know population size. So other than population number, you can also measure population density and geographical distribution. But for the IB, we're only going to focus on population number, all right? So it's easy. When I ask you, go count the number of individuals in that specific population there, you'll be like, oh yeah, it's super easy. I'm just going to go there and count them one by one. I'm going to count each individual. But then you realize when you get there, oh crap, there's like 1 million freaking individuals in this population. Counting each individual is just not possible. It's so time consuming and difficult. And not only is it time consuming, a lot of organisms, guess what? Look here, are motile. So guess what? Try and count the amount of penguins in here. You're going to be like, oh, count 1, 2. Oh crap, I already counted that one. What about, oh no, oh no, that one's hiding behind that one now. You're going to see, it's just a nightmare. You're going to like completely forget how many you counted, and your number will be completely off, right? So for many reasons, it is not good to count each individual, which is why scientists now, what they do is they count in estimates. So if this is the entire population, let's pretend there are millions of individuals in this population. So what scientists will do now is they will go and take different samples. So they will maybe count the total amount of individuals in this square here. And then based on that number, they will do math to extrapolate. If there are this many individuals in this square, then mathematically, how many individuals will be in this full area? And then they will get an estimate of the amount. And it will be mostly accurate. It won't be entirely accurate, but it will be mostly accurate. And it's going to be reliable enough for most purposes. All right. Now, the samples they get, okay? Because you have to take samples, right? And the more samples you take, the better. Just now when I was talking, I was explaining with just taking one sample. But a lot of scientists take multiple samples across the area, and then they extrapolate to find the full number, the real number, to in order to make the number more accurate. Because the more samples you take, the more accurate your estimate will be. If you only take one sample, then it might not be accurate because maybe this area that you took has the least penguins, and actually they're all located up here. And then if you extrapolate, then your answer will be wrong. So the more samples you take, the better. Now, the samples you take can be random or systematic. Random is when you choose arbitrary zones. So you can ask a random number generator to tell you which zones to count to make it as random as possible. Or you can do it in a systematic way. So, for example, I'll show you here. If this is the area you're trying to count, you can do it systematically where you count, for example, maybe every second square or blah, blah, blah. Or you can do it very randomly where it's truly random like this. So both of these are different ways of taking a sample, and they both have their own advantages and disadvantages. This one would be a bit more, actually we're going to talk about it on the next slide, all right? So this is the big idea I want you to get here. Now, when we count population size, we're going to use different methods to count sessile organisms size than we would if we count motile organisms. Okay, so on the next slide, we're gonna talk about how they count sessile organisms. Sessile organisms are just those that cannot run or move anywhere. They're fixed like plants, whereas motile organisms can move. And the method we use to calculate their population sizes will be slightly different. So now I'm going to show you how to sample sessile organisms. So here we have our little population of daisies, and we want to count them, but we don't want to count them one by one. We want to take some samples and then extrapolate so we can know the whole population, an estimate number, right? So what we're going to do is we're going to do random or systematic sampling, like I just talked about on the previous slide. And to do that, you need a quadrat. So a quadrat is the little square thing I just showed you on the previous slide, a square with a particular dimension, and the dimension depends on how big your area is that you want to measure and how big your organisms are that you want to measure, right? So here we have our quadrat, and you're going to place it over the area you want to measure. So essentially what it's doing is it's evenly dividing the area that you want to count into equal size blocks. Now, you want to do either random or systematic sampling. I'm going to choose random, which means I'm going to randomly choose certain blocks that I want to count in. Okay, let's say I choose those 5 blocks. So what I'm going to do now is I'm going to count the number of daisies in each of these samples. Okay, let's do that. So we can see here in this block there are 2, this one there are 3. We count a daisy if it's more than 50% inside a block or a quadrat. Okay, and here we have 4, here we have 3, here we have 3, here we have 3. So now we add up that number that we sampled. Okay, so that's going to be 2 plus 4 plus 3 plus 3 plus 3. That's 15. And how many little blocks did we count in? How many quadrats? 5. So we divide that number 15 by 5 because this will give us the average number of daisies by average per square here, per quadrat. So that will be 3. If you do the math here, it's going to be 3. So what we're getting is, on average, one square here has about 3 daisies, because this one has 4, but that one has 2. So by average, they have 3. All right? So we know each quadrat by average has 3. So now that we know that, we know there are total 25 quadrats, 5 by 5. So if we know each quadrat by average has 3, then we can times that by 25 to figure out how many daisies will be in this big space. All right, and that will give us 75. So see, that's exactly how it's useful. It saved us a lot of time. Otherwise, we had to go and count every single quadrant, and that's useless. And this example, it's not that clear how useful it is, but imagine this area had millions of daisies or millions of other organisms. Then counting would be really redundant. So then you really need this kind of system. I'm going to give you an alternative scenario and see how you respond to it. So now look at the distribution of the daisies. If we were to use random sampling now, if we only took a few random samples, what would happen? Let's say your random samples were from here, here, and here. If you extrapolate the final number of daisies, you will have a very low number, because this area where you took samples from by random sampling was very low. So when you extrapolate it, your total number of daisies will be very low. The same if you do the opposite. If you take a sample from here, here, and here, and you extrapolate the number, then your number will be very high compared to what is the truth. So this is an example of sampling error, when the reason why your final number does not match the real number is because of the way you took samples, all right? So in a scenario like this, because maybe the reason why it's like this is because the ocean at high tide spills a lot of water onto this sorry, I'm dying here. Spills into this area, preventing daisies from growing in this specific area. So in this scenario, the better thing to do would be to do systematic sampling, where you take samples every couple of meters for the whole area, because that way your final answer will be more reliable. Because in this case, there's clearly a gradient. There's more daisies here than there is here. So the more you go this way, the less daisies there are. So in a scenario where there's clearly a gradient in the population, it is better that you do systematic sampling. That will give your final extrapolation a more accurate and it will give it more reliability given the circumstances. Now we're going to talk about the slightly trickier one, sampling motile organisms. Remember before, when I asked you to count the number of penguins when they were all moving about, it was freaking impossible, right? Yeah, so we're not going to be able to use the same method as we do for sessile organisms. We cannot use the quadrant method because if you try and count a quadrant, the number will continuously fluctuate. Maybe 1 second there's 1 penguin, the next second there's 10. You're not going to be able to do it. It's just not feasible. So we're going to have to use another method. And this method is called the capture, mark, release, recapture method. I love this name because it literally tells you the steps of what you need to do. So we need to use, along with doing these steps, we need to use an equation. And this equation looks like this, okay? Don't worry about it right now. It will make sense as I talk about it. I'm just revealing it now. And it's called the Lincoln index. I hate that word. I don't know why there's an L. Lincoln. So this will make sense as I talk about it. So our first step is, here's our population, is we need to take a sample. And we can't take it using the quadrant method. So we're just going to take a random, we're going to go there and we're going to capture a few of them. Let's say 5. Okay, we capture 5 of them. And now what you need to do is count them. How many did you capture? 5. That is going to be our first number here, M, the number of individuals caught and marked initially. Okay, so our population, total population will be M times N over R, right? So our first number is going to be M. That's the number we catch initially. This is the sample that you take. Then what you need to do is you need to mark these organisms. You need to find a way to recognize that you've caught them. So if you catch them again in the future, you know, oh, I've caught that one before. So that's called marking. And marking can look anything like this. It depends what organism you're using. It can be putting bands around the feet. Maybe you've seen this on birds, putting a collar, putting an ear tag, putting paint, putting numbers, anything like this. All of these classify as marking. And it's simply a way to identify which ones you've already captured. So let's say we've marked them now. Okay, so here we go. We've marked them. So I'm going to change their color here for you guys. Now what you need to do, so step 2 was marking, by the way. Now what you need to do is release them into the environment and let them remix, rekindle, become cozy with the rest of the population again. So you want to release them and you want to let them have a fair chance of mixing. And we're going to see why that's important shortly, okay? So just know you need to make sure that they mix. They mix very well. And this can take a couple of days or a couple of hours, preferably the longer the better. And you let them mix. Let them rekindle with the population. And now you need to recapture a sample. Recapture. So let's say now you recapture a sample. That's the last step here. Okay, so let's say now we take a sample of 4. Okay, so it doesn't have to be the same size as the sample of the first time. You just need to take another sample from this constantly moving population. Now we have 4. So that's going to be our number N here. N represents the number of all individuals recaptured. So we recaptured 4 individuals. So that's going to be 4. And now, what is R? R is the number of marked individuals recaptured. So out of the people we recaptured, one of them is marked. So that's important. So you can see why marking is important, because otherwise we wouldn't know which ones we already captured. So if we do now the math, 5 times 4 is 20. So we're going to write this equals 20. I think I already did this, to be fair. No, I didn't. Okay, 20. So that is now our estimate of this motile population. So we're estimating that there's around 20 individuals in this population using this Lincoln index. And if you really count them, there's actually 22. So you can see this estimate is really freaking close, and it's really useful for a motile population. So now for this concept to work, this method that we're using, we are making certain assumptions, okay? Otherwise, if we don't make these assumptions, then it won't work. The concept won't necessarily work. So our first one is we're assuming the population is relatively closed. So we're assuming that these guys cannot just move to another country, or other population can't just randomly enter. We're assuming that this population is relatively closed. They can move around, but they can't exit the population. Otherwise, it would screw up our math, because if these guys, say all of these guys just freaking left, that would screw up our math, because now we can't recapture any individuals that are marked. And if we can't recapture any individuals that are marked, then our population number will be overestimated, okay? Overestimated. So that's important. It's very important. Okay, the next one here is going to be mixing is complete. We have to make sure we give them time to mix, because imagine you don't give them time to mix. I want to show you what happens if you don't give them time to mix. Say you don't give them time to mix and you take another sample, okay? And that sample is from the same area that you took the first sample. So because you didn't give them time to mix, now you're recapturing let's say four of those individuals, okay? 4 of those individuals again. So now your math's going to change, because the number of individuals that you recaptured is 4, but the number of marked individuals is also 4. That's going to change your math to being something else, to being what? 5 times 4 over 4, that's 5 times 1, that is 5. So this makes you estimate your population as being 5 when it's clearly way bigger than that. So that is why I'm emphasizing that you need to have them mix. Otherwise, it will mess up your math, okay? Make sure you have them mix. Okay, make sure it's equally easy to catch each individual in the population, okay? That's not much under your control. Now, make sure the marks cannot get removed. Make sure they don't just drop off easily or can be removed easily by the organism. Otherwise, when you capture an organism that you caught previously, you will not know, because the label fell off. So make sure that's important. Now, what's the limitations of this whole method? Very easy. We don't like marking individuals. It's evil. What if this one is still growing, or it's getting fatter and now it's constricting its neck? What if this is making them easier to be caught by predators because now there's something shiny on them? All right, so you can see this is the one thing that we don't like too much about the whole idea. And it may injure them. To capture these guys, it will take effort, because they're going to try and get away, and it may hurt them. So that's what I want you guys to know about this method. Here we go now with a couple IB-style multiple choice questions. Let's give them a go. So what is the primary difference between a population and a community? A population includes multiple species. Nope, it's wrong there already. A population is one species. B, a population consists of individuals of the same species, while a community includes multiple populations of different species. That sounds very right to me. I think it's going to be B, but we'll make sure these other ones are eliminated. A population refers to the abiotic environment. No, population refers to the organisms, not the abiotic environment. A population is part of a community, but does not interact with other populations. That's not true. A population can interact with other populations for sure, right? They can. One's population of zebras can interact with another population of lions or zebras or whatever. So B is the correct answer here. Which phase of the population growth curve is primarily influenced by positive feedback? We know it's phase 1 here, this phase here where the growth is exponential. So it's going to be A, because B, C, and D, it's all related to negative feedback. Negative feedback is starting to influence it, all right? So the answer here is going to be A. Now, guys, get yourself a coffee, do some push-ups or whatever you like to do, and then we're going to continue with these things. Next, we're going to talk about intraspecific relationships. Remember earlier in this video, we said a population is a group of the same species. So for example, a group of lions. Now these lions can interact with each other in a good way, in a bad way, in any way, but they can interact with each other. We call that intraspecific relationships. So here we have a little diagram, an environment, and I'm going to give you some examples in this environment. So to help you understand the spectrum of what intraspecific relationships can look like. Now, so to break it down, intra means within, and specific means species. So these are relationships within the same species. So within a group of lions, within a pack of wolves, whatever. Now, relationships is a very, very broad term, as I mentioned. It can mean two things. We can classify relationships into two broad categories. And you'll see On the left here, these three examples that I'm going to show you are examples of cooperation. So relationships that are good, win-win situations. So the individuals will help one another. They'll both benefit from the interaction. Whereas the other one is the flip side of the coin. Competition, win, lose. So one individual gets the benefit of the doubt, okay, and the other one gets the short end of the stick, all right? Or they can both lose. For example, if two lions fight each other and they both lose their limbs, that's a lose-lose situation to me because nothing good happened out of that, right? So you can think win-lose or lose-lose. So we're going to look at three examples. I'm going to run through these because you can think of your own examples. You don't need to confine your mind to these. Just make sure you know some examples for the test. It can be your own examples, right? So let's look at a lion. You know, lions, they actually have a cool relationship where they help each other, where they take turns or shifts of who is watching out and protecting their little home, their environment, protecting the kids, while the other ones can be sleeping. So they'll change this person every couple of hours. That way they can all relax, and now only one is struggling at a time. Only one has to be nervous at a time. This interaction benefits the whole group, so we call that cooperation, and that prevents exhaustion, because if they all had to look out together, then no one would get sleep, no one would be able to eat, and it'd just be unnecessary. All right, another one here is wolves. You know, wolves, they actually hunt in packs, and this is very useful because it increases their chance of success, because if they hunt in packs, they're more likely to exhaust the prey, and because there are so many, they can attack this prey from all angles and reduce the odds of this prey escaping. All right, so that's an example of cooperation, because now they can all eat, whereas if they maybe hunted individually, they would struggle to get food. And now bats, you know, bats, especially vampire bats, they have this cool relationship where basically when the mom goes out to look for food for the kid, they will basically delegate their role to another mother and say, please look after my kid so my kid is safe, so I don't have to worry while I am hunting for food. So this way it's nice, because then the mother doesn't have to stress over her kid that might be eaten by a predator while she's out hunting for food. And she will return the favor when the other mom wants to get food, all right? So these are three examples of cooperation. Now let's look at the competition. You know, we talked before about when trees grow, they start taking up space, right? So let's say this is the same species. And eventually, when it gets so much, they will start to compete, and light will start to become a limiting factor. Soil and space will start to become limiting factors. And in that scenario, now, although they're the same species, now they're competing for those limited resources. And obviously the one that is stronger will survive, and the other one will start to suffer and die. So competition for resources is an example. Also, physical fighting. You know, gorillas, they sometimes fight to determine who's the alpha, because the alpha will have the benefit of being able to find a mate and reproduce to make offspring. So that is physical, hand-to-hand combat. That's an example of competition. Now, so just know, because most people, when they learn about intraspecific relationships, they always think fighting is the main thing. But notice there are other ways that they can have competition other than direct physical combat. Okay, one more example here that I have for you is lizards. You know, lizards, they're ectothermic organisms, which means that they regulate their temperature by changing environment. So if they want to be warmer, they have to go to a warm environment. If they want to be cooler, they will go to a cooler environment. So in this scenario, they always compete for rocks, because rocks are very warm and they can get sunlight and regulate their body temperature so they can survive. If they don't do this, they could die. Now, because there's only limited number of space with appropriate temperatures, when there's many of them, they have to compete for this optimal spot. So, and again, this is a kind of example of competing for space or resources. All right, so these are the examples. I hope they help you. But again, you can use your own examples. Just make sure they make sense and understand that there are these two big categories for intraspecific relationships. Following this, we can talk about interspecific relationships. This is different from intraspecific relationships, because intraspecific relationships was between members of the same species. This is a relationship between members of different species, all right? Interspecific, between different species. Now, this relationship is a very broad term. Once again, it can refer to any kind of relationship, good ones, bad ones, working together, anything like that. And we're going to cover 6 big categories that you need to know. The first one is herbivory, when a heterotroph eats an autotroph, an animal eats a plant, right? That is a interspecific relationship, because these are two different species interacting, right? That's example 1. The specific examples that I'm using of these big categories, you don't need to memorize. You can use your own examples, but just make sure you know a example at least. Next one, predation. This is when a heterotroph eats another heterotroph. So a lion eats a human being. Okay, they are different species and they're interacting. Now, in these two cases, one of them were clearly benefiting. In this case, it was the lion who came from another campsite. And in this case, it was the antelope, right? And the others were not benefiting. Now, in the next example, we're looking at a relationship where both of them benefit. This is called mutualism. We're going to use the example here of a bee and a flower. We know flowers contain a super sweet nectar, a nice food source for this bee. So the bee is going to go there and it's going to get its food. So that's how the bee benefits. Now the plant benefits because when the bee goes there, he picks up pollen, sticky pollen. And when he flies to another flower, he can transfer that pollen to that flower to cause pollination. That is how plants reproduce. So the flower relies on the bee to reproduce. So that is how both of them have some sort of benefit from this relationship. And again, we're going to talk a bit more about other examples later in this video that you need to know for the IB, all right? So the next one we can talk about is interspecific competitions. This is when two different species compete for something, some sort of resource, whether it be food, space, sunlight, anything like that. So a quick example would be this one here. We can see this freaking shark really wants to eat this guy. And this lion, he's also staring at this guy. He wants to eat him too. So they're competing here for the same food source. That is one example. Another one would be this tree competing with this grass for sunlight. They're competing for a resource of sunlight, and they are different species, all right? There are a ton of examples. Now the next two are a little bit tricky to distinguish, but let's see if we can do so. Parasitism. So one kind of relationship that can happen between two species is parasitism. This is when one species benefits at the expense of a host organism, the other species. So for example, the one I want to use is malaria. Malaria is a single-celled organism that can act as a parasite. What it does is it will reside inside a mosquito because the mosquito will serve as the host. And the parasite, malaria, requires the mosquito to survive. How it works is it will suck out the nutrients from the mosquito and use it for itself. So in this scenario, the host has no benefit at all. It is just being leached off of by this parasite. Now, when the malaria becomes mature over a lot of time, eventually the mosquito can bite a human and transfer that mature malaria to you. Now you become the new host. Now the malaria will travel in your bloodstream and will go to your red blood cells. And there they love to reside because there's a lot of oxygen and nutrients for them to survive. And they will suck out all those nutrients and even cause damage to your red blood cells in the process. In some scenarios, the parasite can cause symptoms or disease in you, but it doesn't have to. So the host may or may not be harmed. In the case of malaria, yes, in humans it causes disease and it can even cause you to die. So that's what a parasite is, all right? Now, that's what parasitism is. Now, what is pathogenicity or pathogenicity? I hate that word. This is basically the ability, so you're not a pathogen. A pathogen is like a microbe, like a bacteria or a fungus, and we know these things like to infect us and cause disease. So pathogenicity is the ability of these microbes to infect us and cause disease in different species. Now, the difference between these two is that a parasite requires a host organism to survive. Without that host organism, it will die. Okay, and a parasite does not need to necessarily cause disease. It can, some parasites can, but it doesn't need to. Whereas a pathogen, its whole thing is it doesn't require a host to survive. It can reside in different places by itself and survive. It doesn't need a host. But the difference is it causes disease. A pathogen infects you for the purpose of causing disease. So that's another distinction with a parasite, which doesn't necessarily cause disease. Now, okay, great. So I don't want to go into any more than that because any more would be unnecessary. Now we're going to dive a little bit deeper into predation and prey. You guys have to know something about predator and prey relationships, especially how their population numbers change with time. And we're going to use an example here to illustrate that. The example that you guys should know about is the Canada lynx, which is going to be our predator, and the snowshoe hare, which is going to be our prey. The Canada lynx looks like this. Damn, it's got big feet. And I guess this is an adaptation for it to be able to survive in the snow and work well there. And here is our snowshoe hare. It's got big feet. You can't clearly see it here, but it's got big feet. So this is going to be our prey. All right, so let's follow and see what happens to their populations. So initially in a population, we know by default there are more prey than there are predators, right? There are more antelopes in the wild or more zebra than there are lions, right? That's just default. The higher we go up the trophic levels, the less numbers in their populations there will be. We'll learn all about that in C4.2. So that is the default. The automatic population ratio will be this. Then what happens is, because the prey don't feel much intimidation from the predators, because there are a low number of predators at this point, they can thrive and they can have many offspring and they can find all the resources in the world that they want. So because of this, over time, their population number will increase dramatically because there's not much prey going on, so they can just thrive. Now, following this, now there's a lot of food. So guess what? The predators, remember, predation was a density-dependent factor. So now that this population of rabbits are increasing, these hare is increasing so dramatically, now it's going to get very easy for predation to happen, right? To limit their population size, they'll keep them to their carrying capacity, as we talked about before. So now they're everywhere. So these lynx can eat left, right, and center any hare that they want. So what will happen is, so that's what will happen. Now, guess what will happen? The prey's population will decrease and the predator's population will increase because they are now eating a lot of food. This means that the predator will be able to have sufficient food and be able to have a lot of offspring and family members. So their number will start to rise as the prey's number starts to drop. So you can see in this timeline, the prey's number will peak first, and then after some time, then the predator's number will peak. All right. But now, what's happening is the prey's number has decreased so low. Let me reel some words. The prey's number has decreased so low that the predator have now too much competition to compete for these minimal numbers of prey. So now that they will compete interspecific competition, and their numbers will start to decline because there's only a certain number of food, and they will no longer be able to survive as well. So that will return us to our sort of original situation that we had, and that cycle will repeat. So now we need to look at a curve that basically represents exactly what we just talked about. Let me show you. So here it looks complicated, but it's exactly what we just talked about. On the x-axis here we have time, and on the y-axis we have population size. And initially their population was a little bit different. The prey was a bit higher than the predator because that was by default. And then because of that, the prey's population started to increase and reach a peak. But when it reaches a peak, now the predator has a lot of food, so they will be able to yum 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 yum, and as they're eating them, the prey's number will start to decrease and the predator's number will start to increase until it reaches a point where the predator's number is now very high at the peak and the prey is getting now very low. And when the prey's number is getting very low, now the predator have too much competition, and so their number will start to decline. And now that pattern, and now it resets it to its original pattern, and now it will repeat. So what I want you to take away from this curve is this. Two things. It is important to know that their maximum value is out of sync. The predator always peaks before the prey, all right? No. Predator always after the prey, all right? So that's very important that I want you to understand. I hope this idea makes sense with this example. Now next, we're going to talk more about mutualism. Basically, the IB has 3 examples of mutualism that they want you to know. So these 3 that I'm going to talk about, you have to know for the IB, all right? So these are the 3 examples. Now, the thing is, these 3 examples are a little bit complicated. They're not as straightforward as the example of the flower and the bee. So therefore, we're going to have to talk about these to make sure they actually make sense to you. The first one is going to be this one here, plant root nodules and bacteria. So this will be a mutualistic relationship between certain plant species and bacteria. Okay, and we're going to see exactly how this relationship works. So this applies to plants in the Legume family, or the scientific name, I'm not even going to try to say that. So for example, plants that belong in this family are beans, lentils, peanuts, and clovers. And what happens is this. To understand this, we need to take a look into our root system here. So we're going to open up this pot, and we're going to see inside there, in the root system, there are these bubble sac-like things. We call these root nodules. These are little sacs that grow, and within them resides certain bacteria. And this bacteria is going to be very helpful to these plant species, and we're going to see how. So let's take a look and zoom in to these little root nodules, these little sacs that grow on the roots that contain these bacteria. Now, the specific bacteria that we want you to know is bacterial species Rhizobium, all right? So this is important. So this relationship is happening between bacteria called Rhizobium and the plants and the Legume family. So how does it work? Basically, you know a plant needs various nutrients and food. For example, it needs nitrogen. One of the things that it really needs is nitrogen, or N, from the periodic table. It needs nitrogen because nitrogen is required to synthesize amino acids and proteins because they contain nitrogen, right? And also nucleic acids like DNA and RNA, they contain nitrogen. So that is very important. You need to understand that plants need nitrogen. And usually, a lot of soils contain nitrogen that is usable, because nitrogen, what you need to understand is nitrogen comes in various forms. And some forms can be directly used by the plant, and other forms cannot be used directly by the plant. So a lot of the times in the soil, there is nitrogen that is usable by the plant, and the plant can just absorb it and use it as normal. But some soils do not contain this usable nitrogen source, and so the plant has no way to obtain nitrogen in certain kinds of soils. So for example, in these soils where there is no usable nitrogen, that is where these guys, these root nodules and this bacteria will play a very important role. So we know the environment, right? In the air, there is nitrogen in the air in the form of N2, because nitrogen can come in various different molecules or different shapes. Now, N2 is in the air, and it is one of those nitrogen sources that is simply not usable to a plant. They can't extract nitrogen from the air, all right? They can use nitrogen that's in the form of ammonia and nitrates and things like that, which may be found in certain soils. But in this scenario, we're saying that this source is not in the soils. It is not found there. But not to worry, these plants have these root nodules, and these root nodules contain these bacteria, and these bacteria can convert this non-usable form of nitrogen by a process called nitrogen fixation into a usable form here in the roots. And that allows the plant now to use this usable source of nitrogen in the form of ammonia or nitrates and things like that. And that allows it to be able to make amino acids and nucleic acids now, which is important for its survival. Okay, so we can see now how the bacteria how the plant benefits from this relationship, right? So how does the bacteria benefit? Well, the plant, if it manages to be kept alive, it can do photosynthesis and make a lot of glucose and nutrients like that, which it can send down via the root system to these root nodules where the bacteria reside, and the bacteria can use that for themselves so that they can stay healthy and happy, right? That's important. So this is a mutualistic relationship between this bacteria and this plant, and we can see how they both benefit. And this is very useful because this gives this plant a competitive advantage, because it is able to grow in certain soils where there is no usable nitrogen forms, because it doesn't matter, because these root nodules, these bacteria, can help create these usable forms from the air. So this allows this plant to be at a competitive advantage compared to other plant species, because it can grow in any kind of soil or specific kinds of soil where other plants cannot grow. The second example you guys need to know about involves orchids. So orchids are these beautiful flowering plants, and they have a mutualistic relationship with fungi. So fungi is a kind of eukaryotic organism, right? It could be single-celled or a multicellular organism. Now, what is the relationship between the orchid and the fungi? So you should know that orchids are not that good at making their own nutrients. So a lot of them are not even able to do photosynthesis, and that's actually surprising, right? So they are not very good at making their own food. So the thing is, when they are a single seed, they have a serious trouble germinating and becoming a flowering plant, because their seed does not have adequate nutrients to supply the process of germination and plant growth. So therefore, they really depend on fungi. So I'm going to show you here. Fungi can exist in the soil. And fungi are, like I said, are these eukaryotic cellular organisms. They can be many cells or single-celled, and they can form these branches. Now, these branches are called hyphae, and they can extend everywhere in the soil. And the whole purpose of these hyphae is to translocate or transport nutrients from one place to another under the soil. And in the case of the orchid, when the orchid is a single seed, it needs more nutrients to be able to germinate. So a lot of these fungi can transport nutrients towards the seed to give it more nutrients so that it can germinate. Now, these connections between fungi and the plant root system, or connections between the fungi and the seed, we call this mycorrhizae. So mycorrhizae is a term used to describe the mutualistic relationship between fungi and plant root system. All right, so that's very important. So we know that these fungi provide nutrients and transport nutrients to these orchid seeds so that they can germinate. And not only that, they also supply nutrients to the roots of orchids that are already formed to help provide them with more nutrients and help them with increased ability to absorb nutrients from the soil and things like that. So that is how the orchid benefits from this relationship. So now you might ask, now, what is the difference? I mean, how does the fungi benefit? Because right now it seems like it's the only one doing work here. You're right, it does seem so, but where the fungi benefits is at the end of the orchid's life cycle. When it dies, it will now provide a source of nutrients for the fungi that can decompose it and use it to survive and grow itself. So for the fungi's benefit kind of comes much later in the process compared to the orchid's benefit, which comes from early in the process of its life. All right, so make sure you understand the key things about this relationship. The last example you guys need to know about is Zooxanthellae in hard coral. So this sounds confusing. So first we got to break down exactly what this is before we can even talk about the mutualistic relationship. So have you guys ever been, if you've ever been to the ocean, you may have seen corals. You know what corals are? They look like this. They're beautiful and colored. And when you touch them, you realize actually you thought they were going to be soft, but they're pretty damn hard. They're very hard. Now, these are corals, okay? Hard corals. Now, when you look really closely at them, really closely, you will see certain structures that look vaguely like this, okay? You see these? These guys are very small, and we call them coral polyps. Coral polyps. So this image right here, you can see this is the coral, the hard part, and then growing on it, we have many of these. I'm making it blue here. This right here is going to be our coral polyp, and you'll notice covering this coral we have so many of these, and they're very small. They can be 1 to 10 millimeters, so they're really, really small. And this coral polyp is the living part. This is the part made up of many cells, and we're going to talk more about what it does very shortly. All right, so now if we zoom in to this coral polyp, you'll see something interesting. Let's say we zoom into this tentacle. So they have these little tentacles, as you can see, right? Now, these tentacles, what they usually do is they try and wave a little bit, and they try and bring organisms into this area here. You see this area? That's like this coral polyp's mouth. So part of its nutrition is by eating little organisms, which we call zooplankton. Zooplankton are just little organisms that live in the water, okay? That's all you guys need to worry about. And they come in here. And they're going to eat them, right, and digest them to absorb food, right? So that's kind of one purpose of these tentacles, to bring them into the stomach area where they will digest them. Now, when we zoom into this tentacle, we're going to see this. So we're zooming into this area here. You can see the most outer layer here of this tentacle is going to be made up of these cells called nematocysts. And these cells serve a protective function. They are responsible for causing a stinging sensation if certain predators try to touch them. So that's their main purpose, for defense. And then one layer below it, we have these cells called, which are a kind of algae. You know algae, these are little cells or organisms located in the ocean that can do photosynthesis. So these are algae, cells that can do photosynthesis. And we're going to specifically call these kind of algae Zooxanthellae. So Zooxanthellae is a kind of algae that we're using in this example. And you can see they're right under that first layer. And their whole purpose, why they're located so near the surface, is because they need to do photosynthesis, right? I just told you, algae are photosynthetic cells that live in the ocean. So they need to be very close so they can capture sunlight, right, to be able to do photosynthesis. And this is going to be very important. So pay attention to these Zooxanthellae. Now, remember how I just told you that these guys can bring in zooplankton. They can eat food, right, and break them down to survive. Now, the thing is, that food is not enough for these coral polyps to survive. That's only half of their diet. Okay, if they only have that diet, they will die. They depend on these zooxanthellae to survive, because these zooxanthellae, they can do photosynthesis, and we know that means they can make a lot of glucose and a lot of food, which they can then send to the rest of the cells of this coral polyp, which can use it to survive. So basically, this is like a little kitchen that helps make food for the rest of the coral polyp, thanks to photosynthesis, all right? So that's very important. So what I want to say here is this. So you can see that the coral polyp, so the relationship we're talking about is between the coral polyp and the zooxanthellae. They are two different species, but they depend on each other for survival. Okay, because the coral polyp benefits because it receives nutrients from these zooxanthellae, which helps them survive. Okay, so remember, this is a different species than the coral polyp. Okay, but it's helping it survive through photosynthesis by giving it nutrients. All right. Now, as long as this coral polyp is healthy, okay, so it's getting enough food, its main job is to secrete calcium carbonate into this area below it. And calcium carbonate is a substance that causes hardening. And this is the reason why corals are really, really hard, because of this calcium carbonate deposition, this framework that is being formed, this calcium carbonate structure, okay? This calcium carbonate skeleton, if you like, all right? Now, then you wonder, all right, so what the heck is the benefit of the zooxanthellae then? What's the point of them? Right now, they're only helping these coral polyps stay alive. So what's their purpose? Well, their purpose is the coral polyps is, remember, it's a nice structure that can defend itself against predators. So it's basically serving as a little home to the zooxanthellae. That's one benefit. It's safe here. It's safe here. Anywhere else, it might be eaten or it might not be able to survive. So here, it's a safe home. Secondly, remember, the coral polyp, all of the cells to survive, they need to do cell respiration, and that makes carbon dioxide, right, as a waste product. But for zooxanthellae, carbon dioxide is not a waste product, right? Carbon dioxide can be used by these cells to do photosynthesis. So that is another benefit of living with these guys. It's a source of carbon dioxide. Okay. And some other minerals. That's not too important though. So now you can see how both of them benefit from this relationship. So now one more thing that I want to say is this. I'm going to move this a bit smaller. You notice how corals are very colorful. Now, why is that? It's actually because of the zooxanthellae. They can have different colors depending on which zooxanthellae we're talking about. Sometimes red, sometimes green. But they are the reason why corals don't appear white. You know, when you look at them, they're very colorful. If the zooxanthellae were to die for one or other reason, and a very big reason why they struggle to survive sometimes is because of the ocean acidity. You know, because of increased pollution in the atmosphere, a lot of carbon dioxide is depositing in the ocean, causing ocean acidification. That can cause these zooxanthellae to struggle to survive because the ocean is getting too acidic. Now, if they start to die, now this coral, the whole coral, first of all, the coral polyps will die, because remember, the coral polyps depend on zooxanthellae to survive. So if this whole system dies, now it will just appear white. And guess what we call that? We call that coral bleaching. Coral bleaching. Now they appear white because now we only see the calcium carbonate skeleton. We don't see the coral polyps anymore, and we don't see the color anymore because the zooxanthellae that are responsible for creating the color are gone. All right, so that's very important. That's an important thing to understand here. So I'm going to reveal some other keywords if I miss anything for you guys. So just know that Zooxanthellae is a kind of algae. Specifically, it's under the phylum of dinoflagellates. It's not too important, just be familiar with that word. Okay. So yeah, I think that's all that I want you to understand. Just make sure you understand why there's a mutualistic relationships between coral polyps and these Zooxanthellae, all right, and how they each benefit. Now I'm going to dive a little bit more into interspecific competition. We already said this is when two different species compete for things like resources, land, food, anything like that, right? Now, there's a little vocab word that we need to learn. It's called allelopathy, and you'll see why this is so relevant to interspecific competition. So, allelopathy is a phenomenon whereby certain organisms, like plants or fungi or whatever, they can produce certain molecules that serve the purpose of slowing down or killing their competing species, the competing people, the organisms that they're competing against. Thereby, it gives them a competitive advantage to get to the resources or get to the land and survive better than their enemies, all right? So that's what allelopathy really is. Now, these molecules or metabolites, metabolites, this word, is a very broad term, and we can split it into two categories. Primary metabolites, these are not related to allelopathy. These are just molecules that are functioning in our normal growth and development as organisms. We require them to survive normally. Now, secondary metabolites, these do the opposite. They're basically little metabolites or molecules that are made for the purpose of impeding, slowing down, or killing other species or your enemies, and thereby giving you this competitive advantage. These are involved in allelopathy. So let's take a look at an example of a plant here. This plant is called garlic mustard, and it is a very perfect example to demonstrate this point. What it does is it makes a secondary metabolite called sinegrin. Okay, sinegrin. Now, this guy is actually the reason why wasabi has that zingy taste that it has, and same with mustard. You know, mustard and wasabi, they have that like stingy, zingy taste. It's because of this guy. Now, this thing actually has far more purposes than just taste for us humans. It is an example of a secondary metabolite, and what it does is when it's released from the roots, it will now diffuse here in the soil, and if there are any nearby species of other plants that are trying to grow and establish a root system, sinegrin is going to impede and prevent that from happening, thereby preventing other species from establishing a home here. And because of that, it gives sinegrin an advantage, because now it has a lot of space around it to allow it to propagate itself and make more babies and deliver more seeds to be able to thrive in this area. So that's very important. Not only does it prevent other species that are already there from growing, it can also prevent seeds from germinating. So if sinegrin is present here, if a new seed lands here from another species, that seed will not be able to germinate, because now sinegrin is preventing it from doing so, because sinegrin influences the conditions of the soil, making it not favorable for other species seeds to germinate. All right, so that's very cool. And the cool thing about it, even better, is that it doesn't influence itself. So it's really only influencing other species and thereby getting a competitive advantage. Now, what I want to say also is that sinegrin is also released from the leaves. And in that case, it doesn't really have influence to other species, but it rather has influence to organisms trying to eat it, because it has that zingy taste. So it deters certain herbivores a bit. Okay, so that's our first example here. Now I want to talk about the next example, which is going to be in certain fungi. We're going to talk about microbes, little tiny things we cannot see. An example that we're going to use is the single-celled fungus called Penicillium rubens. And it's actually very interesting how this phenomenon was discovered, but that's a story for another day. Just follow my idea here. So here we have a fungus, and it can be a colony, so it can be many of them, many single-celled ones aggregating together. And what's interesting, this is a petri dish, and let's say we have a fungus, this fungus in here, and there's a lot of bacteria here. We can't see them because bacteria are so tiny. We cannot see them with our eyes, right? We need a microscope. So let's say we zoom in, and what we can see is we can see now all the bacteria surrounding it, and we can see our fungus here, but we can see a clear zone here that has no bacteria. And we will wonder, why the heck is that? Like, do they just not like this guy? Are they just like afraid of it or something? Then what scientists realize, is that actually, this guy makes a molecule called penicillin. And we actually know now, we actually use penicillin nowadays. That's why you might recognize this word. We use penicillin. It's an antibiotic. And an antibiotic is a molecule, or in this case, a secondary metabolite, that can inhibit the growth of bacteria. So we use it when we get bacterial infections. And we actually used it after discovering this idea. And it was actually an accident. But like I said, it's a story for another day. So this is useful for this specific fungus because it creates a clear zone here that allows it to be able to extend or increase its colony size, so the fungus can duplicate and increase their size. So it creates a zone to allow them to increase in size. And also it creates a clear zone where there are no bacteria, so that if there are resources here nearby, then it has access to them more than the bacteria. It prevents the bacteria from getting access to the resources and thereby giving it a competitive advantage. So these are just two examples of secondary metabolites. So we got penicillin here for microbe example and synegrin here for plants. There are obviously many more, but the IB wants you to know these two. So make sure you know these two. And that's all I want you to know about allelopathy. Next, we need to address an important thing about interspecific competition. Sometimes it really appears to us like two species are competing when in fact they are not. So you have to be very careful before you actually decide on that. And as a scientist, you need to take three sort of approaches, or you need to take different approaches to answer this question. So the initial approach is how everyone starts, right? You go and do field observation. You look in nature and you notice something, and you notice a pattern, and you're like, maybe this is the case. So if we use this example here, here's the ocean, and this is like a big rock, and we have these species of barnacles growing on it. So this is one species, and this is another one. Barnacles are basically, I like to think of them as in Pirates of the Caribbean, you know, this guy, how he has all this stuff growing on his face. These are kind of like barnacles. Some of them are barnacles. Little species that can grow on rocks and things like that. It's kind of like a shell, but it's not. You guys can search about that more in your own time. Now, in the field observation, maybe you notice, oh, actually, I think these two are competing because look, they're not growing in the same area. This one is growing here and this one's growing here, but they're not mingling with each other. So they must be competing. So you make that observation, but this observation may not be entirely correct. And I'll show you why. So you're going to construct a presence-absence matrix based off your observation. And what this is this. You're going to have all your sites. So you're going to split this area that you're looking at into different regions or zones like this. So here we have site A, site B, site C. You split it up by an imaginary line. And then you write whether or not the one species is present in that site or not. And if it is, you give it a 1. And if it isn't, you give it a 0. So even if there's a lot of them present, you still only give it a 1. You don't write the amount of them present. You only write 1 if they are present or not. So you can see here for species 1, they're present in site A and B. And then, so in site B, they're both present. And then C, D, and E, only the other one is present, right? So we can see this, and this is our observation. And from this observation, we kind of think, oh, there must be competition, that's why. But actually, a lot of the time, it isn't because of interspecific competition. There is another thing that could potentially explain the reason why it is like this. A reason that is maybe unknown to us at the moment. And I'll show you exactly what I mean. So that brings us to another thing that a scientist can do. So one is field observation, but to get the real answer, they need to do something else. They need to experiment. So they're going to do field manipulation. So in this example, what the scientist decides to do is he's like, well, I'm going to remove this species, this one, and I'm going to see what happens. So he removes it. And he gives it a long time to see if anything changes. And he noticed, actually, surprisingly, that this species here, this species of barnacles, didn't grow into this area. It just remained here. So he may have been wrong because he thought, oh, well, I really thought that they were competing. And I thought if I remove the competitor, then this one will now grow into this area and grow here. But he was wrong. So this shows that interspecific competition is not always the reason why it could appear like there's competition. Maybe some other factor like nutrition in this area is not right for this species, or some other factor like predators or something, any other reason, right? And then interestingly, he was like, so I gotta now be a real scientist and test the opposite thing. So he removes now the bottom species, and he notices something. Okay, and he gives it time. And he notices, oh wow, something very interesting happened. They did grow in this area. So this suggested that the blue one was in fact a competitor to this lighter colored one, because when the blue one is present, the lighter color will not grow in this area. But as soon as we remove this competitor, then it starts to grow there. That infers that they were competing for this area here, and the blue one was kind of preventing this one from growing there because it is a better competitor. It has some advantage that we don't know about, right? So that's very interesting. So from this experiment that the scientist did, this field manipulation, he got these two conclusions. One of them, in one scenario, it showed that there was competition, and the other one, it showed that there was perhaps no competition. Okay, so that's very, very cool. That's the point I wanted to show you. And this brings us to the last bit, which is laboratory experiments. So to find out exactly why the dark species didn't want to grow up here, you need to do experiments in a lab where you can control all the variables. You can control nutrition, you can control the predators, you can control the water levels, you can control everything. And by doing that, by using the scientific approach, you can figure out reasons for why things are happening, why it was, because we know maybe it wasn't interspecific competition. So what is the reason then? So you need to go that one step further and do laboratory experiments. So I really hope this example clearly demonstrates that you should always investigate interspecific competition and not just assume that two species are competing because it looks like it. Let's talk about endemic and invasive species now. So there's a couple of key words that you need to learn from this part, but it's not too hard. So an endemic species is one that is exclusively located in one environment. Basically, it is found nowhere else in the world, or in very few other places perhaps. So that's an endemic species. Now, an invasive species is kind of like the name says. It's a species that usually doesn't live in a specific place. So it's non-native. It doesn't live in that place, but it goes and invades that new area. And when it invades that area, what makes it invasive is that it is actually very competitive, and it can really compete and make life very difficult for the endemic species there, causing a lot of them to die and this invasive species to overtake this environment. That's an invasive species. So we're going to take a look at an example here. That's very cool, I think. Let's just start with it. So you know the Galapagos Islands. Charles Darwin frequently went there for a lot of his experiments. And on the Galapagos Islands, an endemic species to that island is the Galapagos tortoises. And they're very big. And they live here because a lot of grass, and that's their primary diet. Now, the thing is, Charles Darwin, by the way, doesn't look like that. Okay, he looks like that, but I think for the sake of everyone's entertainment, we'll put him. What happened was he noticed by going there frequently that the endemic species, the tortoises, their numbers started to decline, and he was wondering why. And he came up with two answers. And he was thinking, okay, it must be because humans are eating them. Because at this time, people were living there, and it is a good source of meat. People need to eat, right? Need to survive as well. So he was thinking maybe it's that. But then his second thing was he realized actually it could be because of this invasive species. And which specific invasive species are we talking about in this scenario? It's going to be goats. So goats were not endemic to this area. They in fact lived in other places where people wanted them to be for breeding purposes, for farming purposes, or whatever, but they were not endemic to this area. But what happened was the people here started to transport goats. They started to transport the goats to the Galapagos Islands for many reasons, mainly because farming and a food source, right? Because goats can give us some meat so we can eat and give us some milk. So a lot of the goats, the problem is, they weren't kept on a farm. They started to go wild, and we give that a name. When an animal goes wild, or when an animal is now in the wild population, we call it a feral population. So feral really just means wild. So some of the goats escaped the farms and became feral populations, so wild populations. So now they were kind of mixed among the endemic species, the tortoises that live in the wild. And the problem is they're a bit faster, right? So they can eat much more quickly than tortoises can. And not only that, they are new to this environment. So in this environment, there are no predators that target our goats. So their numbers are just going to amplify and increase because nothing's eating them. There's no predators for the goats. There's only predators for our tortoises, right, to keep the ecosystem in balance. But now that we have a new invasive species, there's nothing to eat them. So they're going to increase very fast because of both of these reasons, just because they're simply faster and there's no predators. All right, so that is an example of how an invasive species can cause a problem. So the people noticed this, and obviously they started to try and fix this problem. And so what they do is they did culling. Culling is just when you mass kill a certain species for the sake of the big picture here, which was to save the other animals that were now being influenced by this invasive species, okay? So that's the big idea, and that's an interesting example that you can know about endemic and invasive species. There are other ones, but this is just one example that you guys should be aware of. Finally, guys, you made it. We're at the last segment, chi-square test, and you will be wondering, why the heck are we doing math in biology? Just why? Why, man? I didn't sign up for this. Don't worry, this is actually very related to the topic that we just learned, and I hope it will make sense with this explanation. So if you're an HL student, you guys learned another use of the chi-square test when you guys learn about inheritance. Now we're going to learn about a different use of the chi-square test in this topic. But if you're a standard level student, then this is the only kind of chi-squared application that you need to know about. So another name for a chi-square test is test of association or test of independence. It's a mathematical test that we use to see if there is an association or a relationship between two variables. All right, so a test used to measure the expectations compared to the actual observed data. This will not make any sense to you right now, so don't blame yourself. We're going to use an example, and then it should make perfect sense. So here we have an environment, and we have two species, a clownfish and an anemone, right, from Nemo. These are the two species, and say we want to see if there is a association between the presence of these two species. Are they more likely to be found together or more likely to not be found together? This is our question, and we need to determine the answer to that. Maybe in this example it's very easy because you can see, well, obviously they're more likely to be found together because they're present way more together than they are separately. So that's the answer, right? True. So in this example, you can very easily see it with your eyes. You don't even need math. But sometimes the situation you're given is very tricky. And when you're looking with your eyes, you'll be like, oh, I'm not sure. It could go either way. And for that scenario, you need to use math to determine what the answer is. Are they associated with each other or are they not associated with each other? So for that scenario where it's tricky, we really need the chi-square test. So to move forward, what we need to do is first collect our data, our observed data, going into nature and finding the data for ourselves. That's what we do. So we construct a little table to present our data. So here we have our table. At the top we have clownfish present and clownfish absent. And on the side we have the other variable that we're comparing it to, the anemone presence. So every time a clownfish is present and an anemone is present at the same place, we will write that, we will record that. So we can see in this scenario, there were 28 occasions where both the clownfish and the anemone were present in the same place. And then we fill it in for the rest of the data, right? The rest of the scenarios. And we can add up the total. So 28 plus 2, this will be the total number of times the anemones were present. And this will be the total number of times they were absent. And you do it the same for the columns. The total number of time the clownfish was absent and total number you know what I mean. And then you add up the full total. So the full total will be the total number. You'll add up these two and that'll give you 50, and it should correspond to adding these two. Adding these two up will also give you 50. All right, great. So now we have our observed data, but to do the chi-square test, we need expected data. We need to calculate our expected data. So how do we do that? Let me show you a little example. Let me show you a little example here with a dice and flipping a coin. These are two separate events, just like we had two separate species, right? Now, these events are independent. We know that the outcome of a dice does not depend on the outcome of the outcome of the coin flip does not depend on the outcome of the dice, right? They are independent events, all right? Now, so if I tell you, if you can throw a 6 and a head, you will get $1,000. Then you should say, and then I ask you, so what is the odds of you succeeding? Well, to figure that out, we need to write the odds of the two events separately. What's the odds of throwing a head? 1 in 2. What's the odds of throwing a 6? 1 in 6. So we can write that down. So it's going to be 1 in 6 of throwing a head and 1 in 6 of throwing a 6 and 1 in 2 of throwing a head. So the odds of those two events occurring together is when you times them together. So 1 in 6 times 1 in 2 will give us 1 in 12. So the odds of us doing both of those events together to earn $1,000 is 1 in 12. So that is our expected ratio, our odds, okay? Our odds. So if I say you try 24 times, what is the expected number of times for you to succeed? Well, if you try this 24 times, then because the odds of succeeding is 1 in 12, then you're expected mathematically to succeed twice, right? So that is how we calculate our expected values. So that's just a little example to make it make sense, because it's the exact same idea here. We're going to have to do this. Let me show you. So we're going to calculate now the expected value for each of these boxes. This one here, this one here, this one here, this one here. Because we already have their observed values. So let's do it for this one here. So it's very simple. It works the same way that we just did here. So we're going to calculate now the expected value of clownfish present and anemone present at the same time. So what is the expected value if they are both present in one area? Well, what you do is this. you have to write down the odds of the events happening separately, and then you can multiply them together to get the expected odds. So what is the odds of having a clownfish present? Well, it's very, very simple. Let me show you. The odds of having a clownfish present is 35. 35 times the clownfish was present. Out of how many? Out of 50. Out of 50. Okay, so 35 times out of 50, the clownfish was present, and 15 times it was not present, right? So that's our first ratio. Okay, so that's going to be this one here. Now, then we calculate the odds of the anemone being present. Well, the odds of the anemone being present was 30 times out of 50 because 20 times it was not present. So those are the two separate odds of the events happening. So the odds of them both happening together is going to be timesing those two together. So that's a top times top and bottom times bottom. And then you simplify. So the odds or the frequency of those two events happening together is going to be 0.42. So now to find the total, the expected number of individuals, we have to times this odds, this frequency, by the total amount of, all right, the total amount, which was 50, okay? So we're going to times 0.42 by 50, the same way we did here. When I said, when you throw 24 times, what's your odds of succeeding? It was 24 times 1 over 12, which was 2, all right? So we do the same thing here. So our expected value for this category here is going to be 21. And we're going to do the same thing for all of the other ones. So now that we have our observed data and our expected data from math, now we can actually do the chi-square test. But before that, I want to show you a simplified version of what we just did here. Notice here how we did 30 over 50 and 35 over 50, and then we times it by 50, right? That's the math we did. So in reality, you can simplify this equation by canceling out two of these 50s, this one and this one, because this one is on the top and this one's on the bottom, right? So we can cancel them out and simplify the equation to 30 times 35 over 50. So if you look at the thing, it would be 30 times 35 over 50. So you can simplify the math for you if you want to save time, but if you understand this concept, you wouldn't need to do that. Okay, so now that we have that, now we can do our chi-square test. So here is the equation. This is the equation to calculate our chi-squared value. And to do it, all we need is our observed values, which is O, and our expected values, which is E. With that, we can do all the math. Now notice here in the chi-squared equation, we have a sum, because we're going to have to calculate what's in this bracket for each of these squares, for each of these scenarios. There are 4 scenarios, right? We have to calculate now what's inside this bracket for each of those, and then we have to sum all those values together. So I'm going to show you how to calculate what's in the brackets here for the first box here, but the same concept will apply for the other ones. So here we go. So our first box, our observed value was 28 and our expected value was 21. So observed minus expected, and we're going to square all of that, and we're going to divide all of that by the expected value, which is 21. All right, so that's how you do it for the first box. Then you're going to repeat that for the other boxes, and you're going to add them up together because there's a sum, and that will give you a value. This is your chi-squared value. Now, before we can move on and use this chi-squared value, we first have to make sure that we have a null and alternate hypothesis. So a null hypothesis is a statement where you say that the two events are independent, they are not related. So null hypothesis would be clownfish presence is independent of anemone presence. We're saying that they are not related, okay? They are not related. And then the alternate that's always, the null hypothesis is always going to say that two events are not related, they are independent. That's always the case, so don't mix these two up. Now the alternate hypothesis is going to say clownfish presence is not independent of anemone presence. That is saying that the two events are related. So these are just two opposing hypotheses, and at the end of the day, with this chi-square test, we want to prove which one is correct, or which one we're going to go with. So now that we have our chi-square value, we need to compare it to a value in this chi-squared table. So the question is, which value do we compare it to? So to figure that out, we need to first know what our degrees of freedom is of our experiment. And you're not going to understand what it really means. This is way beyond the scope of this video to really understand what degrees of freedom means. But you do need to know how to figure out which degrees of freedom your experiment applies to. And it works like this. You're going to look at your table that you had, and you're going to do this math. you're going to do row minus 1 and column minus 1. So how many rows do we have? 1, 2, right? We have an enemy absent, enemy present. We have 2 rows. So we minus that with 1. That's going to give us a total of 1. And the same with our column. 1, 2, minus 1. So our answer will be 1. So it'll be 1 times 1, which is 1. So our degrees of freedom for our example is 1. So we know we're going to compare our calculated chi-squared value to one of these values in this row. Now the question is which one? Well, that's simple. In the test, they will tell you to compare it to a p-value, a probability value or p-value of 0.05. Usually, 99.9% of the time, it is going to be that one, 0.05 that you have to compare to. So it's going to be this value here. Okay, we're going to compare our value to degrees of freedom 1 and p-value of 0.05. So this value here. I'm going to talk very shortly about what these p-values mean, but for now, just ignore them. Okay, so our value was what? Was 19.44. So it is bigger than this value we're comparing it to. In the case that your value is bigger than the value we have to compare it to, then we reject our null hypothesis and we accept the alternative hypothesis. And our alternative hypothesis was clownfish presence is not independent of anemone presence. In other words, the clownfish presence is related or associated with anemone presence, meaning that they are more likely to be found together for one or other reason than they are to be found absent of each other or separately, right? And this actually makes perfect sense in nature because these two have a mutualistic relationship. The clownfish resides in here. This is its home. It is protected by this species by living in here. And this species of anemone benefits from this relationship because the clownfish poops and pee-pees here, and the anemone uses that as nutrition to survive. So they have a mutualistic relationships. So it makes sense, actually. It makes sense, actually, that this test proves that they are not independent, that they are actually related to each other because they have a mutualistic relationship and therefore they are more likely to be found together. So as promised, I'll quickly talk about these probability values and why we're using this one. So we're using this one, and what this one tells us is that we are 95% sure that if our value is bigger than this one, then our alternative hypothesis is true. And we're only 5% sure that the null hypothesis is true. If we were to compare our value to this one here in the 0.01 p-value, then if our value is bigger than this one, now we are 99% sure of our alternative hypothesis and only 1% sure of our null hypothesis. So the only difference is, depending which one you use, you become a different percentage of certain or sure of your final answer. That's really what these are. But almost all the time, they're going to use this p-value, all right? Now, if our number was smaller than 3.84, what would have to happen is we would have to say we accept the null hypothesis, not reject. So if it's bigger, then we reject the null hypothesis. If it's smaller, then we accept the null hypothesis. So make sure you are aware of both of those scenarios. For those who are interested, here is another example between gender and social media preference. I did all the math and everything here for you guys, so you can take a look at it, make sure you understand it. I'm not going to talk about it because I don't want to make this video endlessly long for you guys. Okay, so unfortunately, we got to quickly touch on another concept here that the IBs for some reason introduced in this chapter, standard deviation. But it's really minimal that you need to know because it's really a math thing that you guys have to use in math. But let me show you what I mean. So standard deviation describes how data is spread around a mean value. So if we said we interview 2 countries, say we interview 3 people from one country and 3 people from another country, and we asked them how many members are in their family. And then one person said 2 members, the other one said 3, the other one said 10. And the other group that we interviewed in another country said 6, 4, and 5, right? What we can do is we can calculate the mean value. So the mean value for both of these sets of data is 5. So by average, the families have 5 members, right? If you had to take the average. Now, if we calculate the standard deviation using this equation, don't worry, don't freak out. You don't need to memorize this equation. If you need it, they will give it to you. And actually, it's in your calculator, and they will allow you to use your calculator. It's very easy, okay? So now if we calculate the standard deviation for both of these, we'll see that the standard deviation for this one is way bigger than that one. And basically what this shows is that the spread of the data around the mean is much larger. And that makes sense. Look, the spread of the data is really large, 10, 3, and 2. Whereas the standard deviation for this one is way smaller because the data is spread very close around the mean. 6, 4, and 5. That's very close to the mean of 5, right? So that's all that a standard deviation does. So I hope it makes sense. Guess what? You made it. We are now at the end and we're going to do a couple questions and then you are done with C4.1. So in a predator-prey relationship, which pattern is typically observed? Predators and prey populations increase simultaneously. No, they increase at different times, right? There's a slight shift. Prey populations peak before predator populations. Yes, they do. The answer is going to be B. So this one's the opposite and this one is just not true. One more. Which of the following is an example of mutualism? A tapeworm absorbing nutrients from a host's intestine. No, that sounds like parasitism because the tapeworm is using the nutrients from its host to survive, but the host has no benefit. In fact, the host is losing nutrients. It is getting harmed. B. A bee pollinating a flower while feeding on its nectar. Yes, that is an example of mutualism because both organisms have benefit. Two species of birds competing for the same nesting site. No, that is interspecific competition. A predator hunting its prey. No, that is predation. So the answer is B. Okay, guys, that is it for this video. There was a crap ton of stuff, but that is it. And I hope it was useful. And I will see you guys in the next one.